this section is going to be pitfalls in microbiome research, since presumably many of you are here because you're interested in the possibility of doing microbiome research yourself. And so I want to go over some of the places that we've gone wrong, and so have many others, and try to make it so you don't go wrong there yourself. And again, this has been the topic of a number of articles in the field recently, so it's kind of important to be on top of this. So, common challenges and pitfalls in microbiome research. So, starting from the top, the largest source of variation in many studies is interpersonal variation. So if you take a sample from one human and sequence it, who's healthy, and one sick person and sequence them, they'll very likely be different, but that doesn't mean it's because of the disease state. People tend to be really different from each other. So to carry out a meaningful experiment, you have to sample the population much more broadly of healthy people, sample the population of sick people, and then compare them in some more sophisticated way, like the unifrac method I described. All bioinformatic measures are approximate. Much of what I was showing you was statistical, and you have to keep an eye on the approximations you make in order to not run amok. Poor statistics followed by storytelling is common. So just to give you a, imagine again the case of a bunch of samples from healthy people, a bunch of samples from sick people. There are many, many mathematical methods of comparing them to each other, many questions you can ask. And so if you're naive about it, you can just fish through all these different comparisons till you find some p-value that's smaller than 0.05 and proclaim victory on that basis. And you'll find many, many papers like that in the literature. But of course, the more questions you ask, the more chance of finding some p-value smaller than 0.05 by chance. And so it takes some real sophistication to be able to control multiple comparisons, be honest about all the comparisons you made in your study in order to be able to accurately ask whether you had a difference in your samples or not. So to put it another way, it takes sophistication and a lot of spine to be able to do a microbiome experiment and decide there's not much there, because there's going to be so many different ways of comparing. You'll probably come up with something, and you have to be sort of tough to decide that it's really not meaningful. Correlation and causality, this is sort of obvious, but the output from these kinds of studies are correlations, disease state, and a change in the population of microbiota. To get to causality, to get to importance, you often will need to do follow-up studies to challenge the idea that what you found is important. At least making these kinds of associations, the correlations stronger. You can use this discovery and validation cohort approach that Judith and Kyle showed, and others have used also. There are huge and often unappreciated challenges with low biomass samples. So much of what I was showing you was samples that were either fecal samples or oral samples. And those are handy to work with because there's a lot of bacteria in those samples. Now, what happens when you have a sample where there's not much bacteria there at all? For example, the lung washes in healthy people, very, very little colonization in a normal healthy person, if any. So what you see is not nothing. You do a tag sequence, PCR amplify, you'll typically get, at least in some of your samples, some 16S sequence. And that's coming from reagent contamination. We'll have little bits of bacterial DNA in your reagents, from dust, and from other sources. And so it's very important to first figure out if you're in the low biomass sample world, and then try like crazy to figure out sources of contamination, how to model, how to measure your contamination, and then ask if you have a signal over that background or not. And there are many, many papers in the literature where people have confused contamination with something they were hoping to measure, and sort of naively working with low biomass samples. Commonly, there's a lack of absolute abundance measures. For both the tag sequencing and the shotgun metagenomic sequencing methods I mentioned, what you get is a proportion of different kinds of bugs or genes or whatever within your sequence set. That is, what you're talking about is 100%. And some bugs 90%, some bugs 10%, whatever. You're not measuring the absolute amount of bugs that are there unless you do a separate experiment. You could have two samples that look about the same, but they differ by a factor of 1 million in the abundance of the different bugs in each sample. Now, there are fairly straightforward ways of going about studying this. You can do quantitative PCR for total 16S, or for specific target genes that are of interest in your sample, but it is a separate step. It's not something you get automatically. So we're not yet supporting quantitative PCR analysis in the analytical center, but that's something we'll probably want to get to sort of in phase 2 or once we're up and running. Another kind of question is longitudinal instability. If you take a snapshot, if you measure a sample at one time point, how representative is that of a sample from the same person a week later, a month later, a year later? Is the microbiome stable, or is it ricocheting around like crazy? In the gut of healthy adult fecal samples, it's pretty stable. If you wait a year, it may not be very different. It often won't be very different. However, other body sites, vagina, you can see very notable longitudinal changes, even on timescales of weeks. So again, you have to sort of get back and ask, based on what I want to understand, how important is it to investigate this longitudinal component? Is cross-sectional going to do it for me or not? I mentioned circadian behavior, if at all possible, sample at the same time of day. Now, a couple of points about mouse studies. There have been lots of mouse studies published. I think many of them fail to take into account how strong the effects are of caging in mouse studies. So mice eat each other's poop. So all the mice in one cage tend to come to have gut microbiota that look like the other mice in that cage. But they'll often be different from the mice in the next cage, even if there's no other difference between them, even if they're all healthy wild type or something like that. So if you have a mouse study where you have one cage full of control healthy mice, one cage full of knockout mice or whatever, and you sequence and compare them to each other, very likely they'll be different, but it's not necessarily telling you anything about the genotype or whatever. What you need to do is treat cage as an explicit variable in your model, in your statistical model for analysis. Do lots of cages for each condition, although it can be one or a small number of mice per cage. So it doesn't have to be wildly expensive, but you do have to consider the caging variable. And lastly, mice typically start out with different microbiota, which can affect the outcome. So there's the famous story from Dan Littman's lab of the segmented filamentous bacteria, where he was getting different results with mice from different suppliers, and he ran that down and made a bunch of important new findings. So another thing is to keep track of what the microbiota is in your mice, if that might be influencing the outcome of the experiment. Okay, so that concludes a whirlwind tour of the pitfalls in microbiome research, but definitely something to pay attention to.